Hello. Last class we covered protist, and we learned that that's an artificial group. This class, we're going to cover fungi, a group that belongs to Uniconta. So these are fungi or mushrooms, and look at this one on the right. It is a fluorescent fungi that it glows at night, and you can see them in some places on Earth, for example, in the tropics. You can find this type of fungi, which are very interesting at night. And here we have a member of the Amanita, which are a type of hallucinogenic fungi. Are very interesting, and they remind us of the Smurfs if you watch that cartoon. These are the learning objectives for today. So we are going to be able to answer these questions. Which major eukaryotic clade this fungi group belongs to, and what does it mean that fungi are heterotrophic eukaryotes? So that means that they are unable to produce their own food, right? How do fungi gain nutrition? We're going to understand what's a saprobe and the main features that distinguish fungi from anything else. Also, the interactions that they form with other species. And some of the main structures that define fungi, as well as their diversity. This is a monophyletic group. Fungi is a monophyletic group. And these are the synapomorphies. If you don't remember, synapomorphies are characteristics present in an ancestral species and shared exclusively by its evolutionary descendants. So in here, we have the ancestor with all of the descendants. So these Chytridiomycota and other early evolving fungi, as well as Zygomycota, Glomeromycota, Ascomycota, and Basidiomycota, all of them have this ancestor. All of this group is what we refer to as fungi. And this used to be a kingdom, right? So the kingdom fungi. The synapomorphy for all of these species containing this group is the presence of cell walls. The cell walls are made out of chitin. And also they have absorptive mode of nutrition, and we're going to learn what that means. Then some fungi lost the flagella. And you can see here the early evolving clades have flagella. So what do you think made the flagella be, or what promote the flagella to be lost? So we're going to learn that today. But before we explain that, let's go through the different interactions, the ecological interactions that these fungi can have in general terms. So we have saprobic fungi, which are decomposers. So if you forget your food in the fridge, what does it happen? So you start getting this mold, which are fungi that break down this dead matter, this organic matter, and they break it down with some hydrolytic enzymes, and then they absorb it. So this is what happens in the saprobic fungi. So they are decomposer, they are eating things that are already dead, or absorbing them, not properly eating them. In the parasitic interaction, we have a fungi that takes the energy from living organisms. So these are bad guys that attack other living organisms. They infect them, they grow, the mycelia grow. You have to think about the spores that can land in, for example, this end, and then the spore grows, creates this mycelia, the hyphae. It's a structure in the fungi that will grow, and then it will form this protein body. And this protein body will have more spores, or it will produce more spores. So this is the parasitic interaction in which the parasite, the fungi, is going to cause a disease to the host, to this ant, for example. But it can also affect humans or other organisms, other species. And they can be deadly, but some of them are not deadly. In this case, this fungi is a deadly one. There is another interaction called mutualistic interaction that gets established between fungi and, let's say, algae, or fungi and plants, or fungi and ants as well. In these type of interactions, we have a mutual benefit. So the fungi and the other interactor is going to, both of them are going to have a benefit. So it's a positive interaction. So this is the reproductive and non-reproductive structures of fungi. It is very important that you remember that these thread-like structures called hyphae are the mass of the fungi. And now if you think in a forest, so close your eyes if you want, and just imagine the last time that you were in a forest, and under your feet there is a lot of soil, and under the soil we have a lot of these hyphae growing, and some of them are actually connecting plants from roots to the plant to another root through these fungi. These hyphae are going to get together to form this reproductive structure called fruiting body. And this is the hyphae right here. This is a close-up of the hyphae. And what you have to realize is that when we eat a mushroom, we are eating a mass of hyphae all together. So these thread-like structures get clustered here and give rise to this reproductive structure. In this structure, we're going to have the formation of the spores as well. The other thing that you need to remember is this word mycelium. Mycelium is the body of fungus, and that is a mass of hyphae, a mass of these filamentous structures, the hyphae. We have two types of hyphae. One is septated, and that means divided. In this figure, you can see how we have a septum right here or here. This is the cell wall made out of chitin, correct? And we have the nuclei, and each nuclei is divided by this septum. Okay, the other type of hyphae is the kenocytic hyphae or hypha. In this case, we don't have a septum. That means that the nuclei are, multiple nuclei are going to be running into each other in this continuum cytoplasm. And that's what we call kenocytic hyphae. So no septum. Another interesting structure is the haustoria. And these are specific structures associated to parasitic fungi. For example, in this case, we have a plant. These are plant cells, right, with the chloroplast. You can see them right here in the vacuole. But then this hyphal is going to enter into the plant cell and stole the nutrients from the plant. So it's going to suck the sap of this plant or the cytoplasm in particular. And another form of haustoria happens when we have these hyphae, which creates this haustoria, and it can attack nematodes. Nematodes are type of worms, and this fungi is going to attack the nematode through the haustoria right here. Fungus can be asexual or sexual, or they can have asexual reproduction and sexual reproduction. And which is very interesting is that they are going to produce spores, both of them. The asexual and the sexual part are going to produce the spores. So here in this diagram, we have everything in bluish or turquoise. It's haploid. And then we're going to have a form that is heterokaryotic stage, which means that we're going to have two nuclei that are in one cell, but they don't fuse yet. The nuclei do not fuse. And they carry that stage for some time until we have karyogamy, which is the fusion of the nuclei, and that forms the zygote. So before I explain in detail this, I want to emphasize the etymology of these words, because if you learn the etymology, it's going to help you to understand other words in science. So let's go through this. I have introduced the haploid, which means the single set, haploid, one set of the chromosomes, right? And diploid, D2, 2 sets of chromosomes. What about karyon? So all of this comes from the Greek, by the way. Karyon means nut or kernel, and that's the origin of the word nucleus. So karyon. That's why we use the word eukaryota or eukaryotes and prokaryota or prokaryotes. And eu means true nut or true kernel. That means this group that has true nucleus. In contrast, pro means before or ancient, and that's prokaryota or prokaryotes means ancient nut or ancient kernel. That's something that evolved before the true nucleus, the ancient nut or ancient kernel. How about hetero? Hetero means different, and that's something that's going to have, in this case, heterokaryotic means that it's going to have different nuts or different kernels, meaning 2 different nuclei. And you could use the word hetero just to refer that they are different nuclei, but you could also use dikaryotic, which means that you have 2 nuclei, dikaryotic. How about gamy? Gamy means marriage. And let's introduce this one, plasma. That's actually, it means something formed, but I want you to think about this, the word cytoplasm, which cyto is the cell, that's what it means. Cytoplasm is the content of the cell, except the nucleus. Now, we're gonna match these 2 words, gamy and plasma, plasmogamy. That is the marriage of the plasma, the union of 2 plasma from 2 different cells. And just remember, gamete means wife and gametes means husband. Just thought that was interesting. What does karyogamy mean? A, B, C, D, or E? The marriage of the nucleus, which is D. That's what karyogamy means. That's the marriage of nuts, the marriage of nucleus. Now, let's go through the general cycle of fungi. In this case, we have the haploid cycle, and mycelium, which is this group of hyphae, the thread-like structures, are going to develop and form spores. The spores are always asexual, and they can germinate and produce more mycelium, right? More hyphae that all together form the mycelium. But when one mycelia find another one, or one mycelium find another mycelium, there can be plasmogamy. Plasmogamy can occur, and that is the fusion of cytoplasm, only cytoplasm. And this stage, it's going to lead to the heterokaryotic stage. This heterokaryotic stage now has cells that have 2 nuclei. The nuclei have not fused yet. And then the nuclei are going to fuse through this karyogamic process. So when the nuclei fused, the 2 nuclei, that's when we call this karyogamy or karyogamy. The result of this fusion is the zygote. And now we have this stage, which is diploid. The zygote is going to be diploid now. And what process is going to generate haploid spores now? Well, that's meiosis. Through meiosis, we're going to create the spores. And these spores now can germinate and form the mycelium. And the mycelium can either go through the sexual cycle or sexual reproduction again, or can opt to create more spores and germinate and keep function as sexually. So this is quite an interesting life cycle. How about the diversity? These are the main groups of fungi. Chytrids, which have 1,000 species. Zygomycetes also have around 1,000 species described. Glomeromycetes have the less, 160 species. They used to be considered part of the zygomycetes, but now we know a little bit more about this group, and we know that that's a different group. Ascomycetes and basidiomycetes are the ones that are more diverse. Actually, ascomycetes have 65,000 species described. So let's go and learn a little bit more about each of these groups. Remember, all of them share these features: the cell walls of chitin, or made out of chitin, and the absorptive mode of nutrition. And only these 4 groups are gonna lack flagella, whereas these early evolving species are gonna have flagella. One of these early evolving clades is Chytridiomycota, and these are also called chytrids. They are gonna have flagellated spores, and therefore you need to imagine always a flagellated spore needs to be in an aquatic environment or in a humid environment. This is the place where they live. They are mostly aquatic, saprophs, or parasitic. This is one species in particular from the genus Batrachochytrium, which infects amphibians. And in recent years, this parasite, this fungi, has exploded, and it's killing a lot of amphibians and has caused the extinction of several species and the extirpation of several populations of other species. So right now it's an issue, and we think that it could be associated to increase in temperature, so related to climate change, but there are other factors that are affecting the increase of these parasites and also affecting the resistance or the response of the amphibians. So you can think about polluting water, among other factors. What explains the loss of the flagella in derived fungi? So I haven't mentioned this, but based on the information that I presented, you should be able to answer this question correctly. A, an increased arid environment, land colonization. B, non-flagellated fungi lack sexual reproduction. C, increase in water availability. D, non-flagellated spores lack dispersal. E, predators of the spores disappear and therefore they didn't need the flagella to escape. That's very easy, right? So an increased arid environment, land colonization, it's associated to the loss of the flagella. So that is an adaptation resulting from natural selection acting on the species that were colonizing Earth. Remember, from here, so these zygomycetes, glomeromycetes, ascomycetes, and basidiomycetes lack the flagella. So this group, the Zygomycota, that's a division, are also called zygote fungi, and they are abundant in soils. They're gonna have hyphae that lack septum, that is the kenocytic hyphae, and they have reproductive structures in the form of sporangia, that's the asexual structure, or zygosporangia, which are the sexual. Some of them, or few of them, form mycorrhizae. And mycorrhizae is the association between plants and fungi, and that's a beneficial interaction. It's a mutualistic interaction. Myco means fungi, and rhizae means roots. So that's an interaction of plant and fungi, but underground. And you can also find these zygomycota, or zygote fungi, if you leave your bread in the abandoned, your sandwich abandoned, and it will be covered with mold. This is an example of the genus Rhizopus, which create these fruiting bodies as well. This is the kenocytic hyphae. Right? We have explained that they lack the septum, and hopefully here you can see some of the nuclei, and you don't see the septum. So this is how the hyphae look under the microscope. This is the life cycle of this species, and let's look into this. So let's start here. We're going to have a result of meiosis. We're going to have spores that when they germinate, they create a sporangium. And this sporangium, now it's going to be able to form haploid spores. So again, everything in turquoise is haploid. And these spores can go and germinate into this mycelium, and then the mycelium can differentiate and again form this sporangia and release more spores, and it can continue going into this direction in the asexual reproduction. However, these spores can produce two types of mycelia. And in here, we're going to annotate it as type plus and type minus. The mating type minus and the mating type plus are kind of the sexes in this type of fungi. So when one mating type minus and another one plus find each other, they fuse. But they don't find each other randomly. They produce pheromones. And these are volatile compounds that they can sense, and then they can grow to towards each other, and then they finally fuse. This is how they look. This is the gametangia. Once that they fuse, they form this structure, the gametangia, with haploid nuclei. And they, after this, the two cells are going to fuse. And when the cytoplasm fusion, when the cytoplasm fuse, we talk about the plasmogamy. So plasmogamy results in this structure that now has multiple nuclei all living together without having a division, without having a septum yet. Now, this is the heterokaryotic stage, right? Different nuclei living in the same cell. After the fusion occurs, that means the karyogamy, we're going to have a diploid nuclei. So we have several nuclei that are diploid, right? Because we have several nuclei in here. This structure that undergoes karyogamy is called the zygosporangium. So we have the zygosporangium for the sexual reproduction and the sporangia for the asexual reproduction. This is how it works in this species. For example, the Rhizopus stolonifer. And that's the species, the common species for the black bread mold. The third group that I want to present in fungi diversity is Glomeromycota. This is the one that used to be considered part of the zygomycetes, but then recently has been described as a different group. And this happened in 1998. These are exclusively endophytes, and an endophyte is a fungi that is going to grow inside of the cells of other organisms. In this case, we are looking at roots in plants. And in these plants, they can create a mycorrhiza as well. So this is the interaction of the fungi, the glomeromycete, with the roots of the plants, but they create these arbuscules, which arbuscule means little tree. And that's because of the appearance of the arbuscular structure that they create inside of the cells of the plant. The fourth group are the Ascomycota. And we have a lot of diversity here. And of course, yeast is part of this group. They live in marine environments, freshwater, or terrestrial environments as well. Yeast is going to be single cell, but most of them are multicellular. They form multicellular structures. And we refer to these asexual and sexual structures as conidia or ascocarps. These are the sexual structures of the ascus, which is actually the synapomorphic structure of the Ascomycota. Ascus means a sac or a little bag, and this little bag is going to contain the ascospores. That's the spores, the haploid cells. And in here we can see one of these ascos, these sacs containing 8 ascospores. This is one example of the type of Ascomycota. They are called cup fungi because they have this cup shape. And this is the fruiting body or the ascocarp. The carp refers to the fruit of the ascomycete, ascocarp. And if we dissect this structure, this fruiting body, we can see here the asci or the ascus. Yeah, the little sacs. And inside we're gonna see the spores. So this is a magnification of this structure. And again, if we magnify it a little bit more, we can see that all of them produce 8 ascospores. This is one species that I described as a new species to science. This is also cup fungi. It's very tiny. It's like half of an inch or even smaller. And if you I dissected this, you can see the sacs with the 8 ascospores. And which is very interesting about this one is that this fungi associates with pines, and the fungi actually help the pine to survive drought conditions. So it's very beneficial. And if the pines that are undergoing drought, if they don't have this fungi, they can die. So it's a very interesting group. The asexual structure is the conidia, and the conidia has this shape. So we have several cells, and at the top we're going to have the asexual conidia. So the conidiophore is the structure forming the asexual spores in this or the conidia. So the spores are the conidia formed by the conidiophores. This is an aspergillus or in penicillium. And penicillium is going to be forming, or it's one of the ones that is used to extract penicillin, the antibiotic. So the penicillin was first described by Alexander Fleming, and that's why penicillium, this fungi, is famous for. You can see penicillium when you leave your oranges decompose, for example, is one of the fungi that will attack and decompose this orange. And you can see the conidia from this orange right here. In your trip to Asia, the one that I sent you for your spring break, the same way you got a mosquito bite, you got sick after eating in the street, unfortunately. Later on, you found out that you got the Protis gastrointestinalis. A friend of you gives you penicillin to kill the bug. What happens to you after taking penicillin? You recover. A, you recover really fast because penicillin is an antibiotic. B, gastrointestinalis produces penicillin that enhances your immune system. C, you eat the Ascomycota fungi, which produces the penicillin in the natural form. D, gastrointestinalis develops penicillin resistance, a type of artificial selection. E, gastrointestinalis doesn't have a peptidoglycan cell wall, therefore the penicillin doesn't kill it and you remain sick. What do you think is the right answer? Okay, I hope that you selected E because that's the right answer. Gastrointestinalis doesn't have peptidoglycans, right? Gastrointestinalis is a member of the excavata, right, the protist. And they don't have this cell wall made out of peptidoglycans as we found in bacteria. This is the life cycle of Ascomycota. Let's go little by little. Again, in turquoise, we're going to have everything that is haploid. So let's start with our mycelium. This mycelium can form the conidiophores. And the conidiophore, as I was explaining, is going to form the spores called conidia. And this conidia can germinate and form, again, more mycelium. However, sometimes these conidia can run into a different mycelium, a different type of mycelium that has the mating type plus, and the conidia will have a mating type minus. And this is going to fuse. This is slightly different what I explained you before. In the previous example, we have the fusion of two mycelium. And in here, I'm telling you that the mycelium is fusing with the conidiospore, or with the conidia. And this conidia later on is going to go through plasmogamy, the conidia with one cell of the mycelium. These two cells are going to fuse without fusing the nucleus, right? So we're going to have our dikaryotic hyphae right here. And this dikaryotic hyphae now, that is the result of this plasmogamy, is gonna differentiate into this ascus. And the ascus is still dikaryotic. So no karyogamy has occurred until now. Once that the karyogamy occurs, we have one diploid nucleus, just one. Now, we're gonna go through meiosis, and we have the result of meiosis. From one cell, we're gonna get how many? Four cells. However, these ascospores, or these ascus, have 8 ascospores. And that doesn't explain why we have 8 ascospores if we have 4 cells resulting from the meiosis. Well, that's very easy. We just have, we go through mitosis and have these 8 ascospores as result. Then these ascus release the spores, the ascospores, and these ascospores now can germinate, form mycelia, and complete the cycle, and they can go through the sexual reproduction again or to the asexual reproduction. This is what happens in yeast. I told you, ascomycetes are mainly multicellular, but we have yeast, and that's one exception that is unicellular. In this case, we have the ascus, which has the 4 haploid spores, and then these can budding, they have budding and create these haploid structures that they can fuse, they can have plasmogamy, and they also have karyogamy, and they can, now that they are diploid, they can bud again, and then they can have meiosis to form these ascus and or the 4 spores in the ascus. So it's very simple, it's less complex, the life cycle of the yeast, but it's very important regardless of their simplicity in the life cycle. And why is yeast so important to us? Well, we use it in combination to carbohydrates. If we give carbohydrates to yeast, that results in fermentation. And the fermentation is going to produce alcohol and CO2. So that's important in the preparation of cheeses or bread as well, and the winery products or alcoholic products as well, like beer. So ascomycota can interact with algae to form lichens. That's a mutualistic interaction, a symbiotic interaction that benefits both the algae and the fungi. Lichens, hopefully you have walked in the forest or in the deserts. Actually, deserts have high abundance of lichen. Sometimes you see rocks that have this coloration. They can look a little bit yellowish or orange. That's some of the pigments that are coming from the algae or the fungi as well. And here we have the algae layer in this figure. The algae is going to have, as you know, they have chloroplast. Therefore, they photosynthesize. And they photosynthesize, that means that they are going to acquire CO2 and fixate that carbon into organic molecules. So because of that, the algae is being autotrophic. And that's also producing energy for the fungi. But in turn, the fungi is protecting the algae from the environment. So it's mutually, that's a mutual relationship. Not only protects the algae, but also provides nutrients to the algae. These are more examples of the fruiting bodies of the ascomycetes, these cup fungi. And you can see that all of them have this cup shape, and they can have different colors and different forms, different sizes as well. Others can have slightly different shapes, not the typical cup shape. These are the morels, which are edible fungi. And you can eat them in pasta or in pizza, etc. So morels are accessible. Not that expensive, but a little bit expensive. But what's the most expensive fungus in the world? That's truffles. Truffles are so expensive that in some places you can buy black or white varieties at $6,000 or $7,000 per pound. And the reason why they are so expensive is because there is a lot of effort from people hunting. The mushroom hunters have to train dogs or even pigs to smell the fungi, and then once that they detect the fungi, the location, they collect this truffle. So this is Lagato romagnolo, the truffle dog, an expert getting this fungi. Okay, those are the Ascomycota. Now let's talk about Basidiomycota. They are also called club fungi. They are going to be important decomposers, very important in decomposing wood. They can also form mycorrhizae, and this is the mutualistic interaction between the plant and the fungi. And let me emphasize something. Similar as the interaction with in lichens, in the mycorrhizae, the plant is going to photosynthesize and give this glucose or photosynthase to the fungi. But the fungi, in return, is going to give to the plant the nutrients from the soil, such as phosphorus, nitrogen, or other minerals from the soil, and also can have access to water. So remember, those hyphae can grow and grow and grow under the soil and reach areas where water is accessible. So it's a very interesting relationship between fungi and plants. That's the mycorrhizae. The reproductive structures in Basidiomycota are going to be the basidiocarp, and this is the sexual structure, and we're going to see the asexual reproduction via fragmentation as well. But these are examples of Basidiomycota, the mushrooms that we eat, the shell fungi that grow on wood, puffballs, jelly fungi, all of them are Basidiomycota. These are the shapes, the colors, the different sizes. And I don't know what you're thinking, but what you're thinking is not what I'm saying. I'm just seeing only fungi in here. These are the shell fungi growing from the wood. And they tend to be very tough, not as tough as other fungi. So these are examples of Basidiomycota. These are Bluetooth fungi. This one right here, because it looks like a tooth that has been just extracted by a sadistic dentist. Or we can have also coralloid forms in this group with beautiful colors. Some of them can be even larger than this basketball balloon. Now let's learn a little bit about the structures that they form. So in Basidiomycota, we're going to have basidium, and this is the basidium that is going to undergo meiosis, and as a result, we're going to have basidiospores. So one cell in this basidium goes through meiosis and forms these 4 cells in the top of here that are called basidiospores. Hopefully you can remember that in Ascomycota, the older group, we have the spores in a sac. In here, we don't have this sac. These basidiospores are extruded to the outside. This is the life cycle of these basidiospores, the basidiomycetes. And let's start in here. So we have 2 hyphae with 2 different mating types, the type minus and the plus type. When they fuse, they're going to have this dikaryotic mycelium, right? In Ascomycota, we have a conidia. We have this dikaryotic mycelium in here that is the result of the plasmogamy. So the fusion of 2 cells without fusing the nucleus. And that divides through mitosis, right, to form this multicellular structure, the dikaryotic mycelium. After that, after this keeps growing, this mycelium, that's going to form this basidiocarp. And the basidiocarp, it's going to still be dikaryotic. So you see the N plus N means that we have the nucleus is still haploid, but we have two nucleus in these cells. And in these gills, this structure, if you, for example, remember the portobello mushrooms that you have eaten, and in one side of the portobello mushroom, you have a lot of gills. And in those gills, that's the formation of the basidia. And the basidia, as I mentioned before, it's gonna fuse the two cells. I mean, it's gonna fuse the two nuclei through karyogamy, and now we're gonna have a diploid nuclei that goes through meiosis to form the basidiospores. These basidiospores now can germinate and produce more haploid mycelium because they went already through meiosis, right? So they are haploid, and they can go through these fusing them to mycelia again, and now this can complete the cycle. This is how Agaricus bisporus, an edible fungi, look when we section it. So this is the cross-section of the cap. The cap is this upper part that we eat. And in there, we have the gills that I mentioned briefly before, gills or lamella. And in here, we have the basidiospores. And a close-up of these basidiospores. This will look, and you can see in this one, the 4 basidiospores. Basidiomycota are important decomposers. Some are pathogens, such as the wheat rust or the corn smut. Corn smut actually are delicious. That is affecting corn. This fungi can grow on the plant, but you can cook it, and in Mexico at least we eat it a lot in quesadillas. A lot of these Basidiomycota form mycorrhizae, and some of them, as I mentioned, are edible, the Agaricus bisporus, but also shiitake, which has some anti-cancerous properties. Others can be poisonous, such as the Amanita muscaria, also called destroying angel. Others are hallucinogenic and used as with recreational purposes. They are or ritual purposes, more importantly. They can be members of the genus Psilocybe or the Amanita genus as well. These are all edible basidiomycetes. And this is the Amanita. A species in this genus can have hallucinogenic fungi or poisonous fungi as well. And this is also hallucinogenic. This is the other that I mentioned, Psilocybe or Psilocybe. Your roommate has left a sandwich in the fridge before moving out. Now you have to clean the mess. You notice hyphae growing on top of the sandwich in the composting stage. You take it to your biology 204 lab for examination. Under the microscope, you notice black sporangia. After following its life cycle, you conclude it is a blank because it has a blank. What could be? Well, what's growing on bread? It's Glomeromycota? No. Chytridiomycota? No, right? That's the one associated to the amphibians. Zygomycota fungi? Yeah. Zygomycota fungi and Zygomycota have a structure called zygosporangium. So C is the right answer. For this module, I would recommend you to study this table and remember the different clades in fungi and the synapomorphic traits of each of these groups and the ecological impact that they have. For example, the mycorrhizae that they can have in some plants or with some, or in lichens, in the different type of interactions, biotic interactions. So we have concluded fungi. Fungi are a very interesting group that I hope that you have learned some interesting things from them today. Thank you.